the vice president of the Moroccan Society for Regenerative Therapies. With my colleagues, Professor Mohamed Marzouki and Najib Al-Idrisi, we are honored to serve as your host for the insightful meeting. So the webinar of today is organized by the Moroccan Society for Regenerative Therapies Committee, which main objectives are to increase the awareness of regenerative therapies in Morocco and Africa by supporting suitable scientific, clinical, ethical, and regulatory landscapes, hoping to allow an effective and safe application of these innovative therapeutics. We are privileged to have with us an esteemed guest speaker, Dr. Virginie Bartel. We are honored to have her sharing its expertise with us today. So the agenda of this webinar is the following. So after the presentation of Virginie, which will last around 35 minutes, a Q&A session will follow. And thereafter, we will conclude with specific remarks that will be delivered by our colleagues. So we want this webinar to be as interactive and beneficial as possible. So please feel free to share your thoughts and questions. We encourage you to engage with the content and also to make the most of this in a learning opportunity. To you participants, thank you very much for being with us here. And the last thanks and last big thank you goes to the organizing team. Without waiting, I'd like to briefly introduce Virginie, who had led several leadership and operational roles for the development of several innovative therapeutics. I had personally the chance to collaborate with her on one specific cell therapy product that has been developed for the treatment of liver diseases. Once again, thank you, Virginie, for accepting our invitation, and the floor is yours.
Thank you. Good evening, everyone. Well, thank you very much, Mustafa, and thank you very much to the Moroccan Society for Regenerative Medicine Therapy, to invite me today. I'm very glad to be here, to be able to have this fruitful discussion with all of you. So as a quick introduction, I've been working in the clinical operation field for the last 17 years. I've worked as a CRA, project manager, and 8 years ago, I started to work for a cell therapy company where I had the chance to meet Mustafa. Well, I thought that the clinical development for cell and gene therapy would have been quite similar to any kind of clinical development for any other drug. But I realized quickly that it was a all new world and an all new discussion when it comes to cell and gene therapy, because there is specific opportunity and challenge. And the aim of the webinar today is to present some of the challenge I've met, and hopefully we can get a very good discussion afterwards. So here's the agenda of the meeting today. To go through this opportunity and challenges. But first, let's try to have a definition of cell and gene therapy. And there, I'm going to ask you to work a bit with me and use the chat of the meeting to write, when I say cell and gene therapy, what is the first word that comes to your mind? Just write it in the chat. Because I would be happy to see, and it's a good test also for the chat to see if it's working for everyone. So innovation, okay. Maybe Mustafa, do you have an idea also of what word would you associate with cell and gene therapy? The first word that comes to your mind.
it's making a treatment with a viable entity.
Yeah, true. So we've got in the chat innovation, tailored therapy, stem cell, challenging, advanced therapy, multiple using extractions planned, multiple mechanism of action, personalized. And so you can already see that if you have to give a definition for cell and gene therapy, actually everyone has his own definition. And it's probably the first challenge that you are meeting when you work with cell and gene therapy, that the definition is not clear for everyone. Here's some word I have that comes to mind when you think about cell and gene therapy. You think regenerative medicine, you can call it ATMP, it's lead to innovation, it's lead to groundbreaking opportunity. Most of the time it's an opportunity to treat some orphan or rare disorder, but it also there is a lot of work to define cell and gene therapy. Here is one definition I find that comes from the Alliance for Generative Medicine that defines cell therapy, gene therapy, tissue engineered products that are intended to augment, repair, replace, or regenerate organ, tissues, cell, gene, and metabolic processes in the body. There is 4 subtypes of cell and gene therapy. There is a cell-based immunotherapy, like the CAR T cell, like the dendritic cell therapy. There is a cell therapy such as MSC. There is then gene therapy, and there is also the tissue engineered products. So these are the 4 main subtypes and category when it comes to cell and gene therapy. The good thing when it's come to cell and gene therapy, the last 2 decades approximately were a huge increase and a huge growth in the market of these therapies. And the good news is the plan is to be bigger and bigger in the next decade. You can see on this graph what is forecast for the next 10 years, which show that the market and the opportunity of this therapy is going to be very big in the future. However, at the same time, as of today, the market of development for this therapy is still very, very limited. On the graph on the left side, you can see the number of ongoing clinical trials on cell and gene therapy. There is currently approximately 1,800 ongoing clinical trials of cell and gene therapy, which is not a lot considering the number of ongoing clinical trials worldwide. And on the right side of the slide, you can see the approximate number of approved products worldwide, which is also very limited. What is good to have in mind is that among the number of approved cell and gene therapy, many have been approved in the last 2 to 5 years. So it again shows that the market is really growing and growing fast. When it comes to cell and gene therapy, the way to develop this specific compound is actually similar to any other biological drug that comes to the market. Everything starts with discovery and research and development. Then it goes to preclinical research on animals, then clinical research, then the data are reviewed by the authority to give a market approval and for the product to be on the market. And there is a post-marketing study after the approval. So it's quite similar to any kind of clinical development, actually. But when it's for cell and gene therapy, there is some extra challenging. The first thing is related to the product itself. It's, of course, a more complex technology, which is more expensive to produce, more complex to produce at a very high rate. Then most of the time it treats more population because it often targets orphan drug, orphan disease, and rare indication. And it can also treat severe and life-threatening indication. Then for this specific therapy, a very long follow-up is needed. So there is a limited and recent environment which increased number of challenge, because due to the relatively small market, there is a lack of harmonization between the country when it comes to cell and gene therapy. There is some limited provider for the company that can really support and answer the specific need of the sponsor and the company to answer the need of this sponsor. Then a very high expertise is required for this development, and it's a longer development, and of course it will cost more money. So when we target more specifically to clinical research, again, this is the classical pathway when you go through clinical research. You start with a study design, then you have to send your product to the clinical site. The product has to be prepared by the clinical site, and then it's going to be administered to the patient. And all this will be under a very controlled regulatory environment to make sure that the rights of the patient are preserved. First, we will focus on the regulatory framework. And when it comes to cell and gene therapy, well, the regulation is similar to the regular regulation for clinical trial, but there is some extra regulation and some extra requirement when it comes to cell and gene therapy. The first thing is, here on the table, you compare the regulation in Europe versus the regulation in the US. The first thing is that the name itself used to refer to this therapy is not the same in both regions. In the US, you will refer to as cell and gene therapy, whereas in Europe, it will be referred as ATMP, Advanced Therapy Medicinal Product. In the US, you have a framework with multiple guidance. And then in Europe, you have other specific regulation with specific GCP for the ATMP and long-term safety. So it shows that there is a willingness to harmonize a regulation, but that as of today, there is still some discrepancy between the country. However, one of the opportunity actually is this effort to harmonize and to increase the regulatory efficiency. I think the regulatory authority are very well aware that cell and gene therapy brings a good opportunity to treat some specific disease, and it brings an added value for the patient. And they know that they need to improve this harmonization to make the approval and the approval better, and to make sure that the treatment are quicker on the market for the patient. And there is some specific approval pathway that can be used for cell and gene therapy to accelerate the review of the dossier to get a quicker marketing approval after the clinical trial are done. So in the US, there is the fast track and breakthrough therapy designation that are not specific to cell and gene therapy, but the RMAT designation, Regenerative Medicine Advanced Therapy, which is a combination of fast track and breakthrough therapy, is really specific to cell and gene therapy and is aimed to help the company to get a product on the market quicker. In Europe, there is a PRIME designation, accelerated assessment, conditional approval that are not specific to cell and gene therapy, but are there to really help some company to get the product on the market quicker. And in Japan, there is the Sakigake procedure that is also aimed to get a cell and gene therapy on the market quicker. One advice I would give for any company that is developing a cell and gene therapy is really to actively discuss with the regulatory authority since the beginning of development through scientific advices, because the authority are really willing to help the company to grow and to develop the product. Then the next step is the study design. When you go and you have to design your study, your clinical trial, you face also multiple challenges. The first one is the limited number of patients. As we said before, there is quite often the cell and gene therapy target rare and orphan disease, which means that there is a very limited number of patients. And unfortunately, the traditional randomized controlled trial might require a need for a high number of patients. And there is a limited statistical power because of this low number of patients. Then there is also ethical issue in the study design, because traditional randomized controlled trials are not suitable for life-threatening indication sometimes. There is some opportunity also to design the study in specific ways so it can answer to these challenges. The first one is what we call a basket and umbrella design, which allow actually to have multiple indication of subtype of indication in only one trial. It might be very useful when you do an early phase safety study to demonstrate the safety of the product, to have this kind of design. Then later, you can have an adaptive flexible trial design that also aim to facilitate the data collection. All this kind of design have the same kind of objective, which is to maximize the data collected for a limited number of patients. This design needs to be created hand in hand with the regulatory authority, again, through scientific advice to discuss with the authorities to make sure that it meets the authority requirement. Then another challenge is transportation to clinical site due to the product itself. There is some logistical challenge in the transportation phase. For example, in the case of an autologous therapy, first the patient needs to go through a specific process for cells to be collected. Then the cells should be sent to the manufacturing facility and then send it back to the patient to be given to the patient. It can be a long process, and every step of the way needs to be carefully followed to make sure that temperature conditions are respected. Most of the time, it's also a cryogenic transportation, which is not standard of care for many Korea. So it's quite important here to make sure that there is specific process in place with the carrier, and that we work with carrier that have the expertise in cryogenic transportation. Then another thing is that if the cell and gene therapy has to be transported by plane, you have to go through the custom border. And most of the time, cell and gene therapy cannot go through X-ray. So it means that a specific custom regulation needs to be applied. And if you do not work with a carrier who's really well aware of the process, it might be a real issue, because it might impact the safety and the quality of your product. Then you have a short-term delivery, because most of the time you have to be very quick between the time the cells are prepared and the time the cells are administered to the site. Then another topic is the storage itself. As I said just before, most of the time it's cryogenic storage that is needed, and the clinical sites are not equipped with a cryogenic tank to be able to store the cell. So it might mean that a limited number of sites can participate if they have enough storage capacity, or you have to find alternatives to be able to deliver the cells and the product to the clinical site. Now, this is a picture, and I want you to, I'm going to ask you to use the chat again, just to tell me, what do you think this is? This is a truck, obviously, but what do you think of the specificity of this truck? Mustafa, you are not allowed to answer to that yet. Clean room? Yeah, any other idea? No? It was actually the first approved GMP approved truck. And it came out of the mind of the previous company, Prometria, who had the kind of issue, logistical challenge that were discussed just before. And with an open mind and a new way to think about it, they created this truck that was approved, GMP approved, and was able to drive to the clinical site. And the cells were actually prepared just hour before to be infused to the patient. So it was really on demand. And the truck was traveling a long way to be close to the site and the patient. And what I wanted to show you here is that I think in cell and gene therapy, there is some specific challenge, that's for sure, but you have to keep a kind of an open mind, because you can come with some creative solution that can answer to a specific problem at a time. Then another challenge is the preparation of the product itself by the clinical site. Again, most of the time, prepare cell and gene therapy is not part of the clinical routine for the site. So it's something new. They're not used to it. It requires some specific training. And for the site, it's also some specific process to learn, to receive the product, to assure the traceability of the product, and to administrate the product to the patient. So of course, the sites are fully able to do it, but they need sometimes some extra training to overcome the fear that you might have when you do something new. And so it's required for the sponsor to show very strong support to make sure that the site is trained, but also supported enough to prepare the product. Last but not least is the patient. One of the challenges is that before to receive cell and gene therapy, for a patient, it can be a very, very long journey. Here, it's a journey for a case of an autologous setting, but most of the time, so the patient is treated with standard of care, because most of the time you don't use cell and gene therapy as a first intent. So the patient goes through the standard of care, and then if it's not good enough for the patient, then the patient might be referred to a treatment center that is allowed to infuse the cell. Then the patient needs to be checked for eligibility criteria. And if, of course, he agrees to participate, then he can go for the first step of an autologous, more often the leukapheresis, to collect the cells. Then the cells that needs to be sent and manufactured into manufacturing facility. Then the patient needs to be prepared to be able to receive the prepared cell, which means that before the patient eligibility and patient preparation, in case of very severe disease, a very short-term disease, this time might be too long, unfortunately, for the patient. Then there is a cell infusion itself. And afterwards, for cell and gene therapy, there is a specific long-term follow-up of the patient that needs to be done for very, very long time, sometimes up to 15 years, to make sure that there is no safety issue for the patient long-term. So you can see that for a patient, before the time he's started treatment for its disease and until he gets a cell infusion, and even after, it's a very, very long journey. Plus, the access might be very challenging for the patient because most of the time they need to go to specialized treatment center to receive this product. So it's a limited access for the patient. Not all the patient lives nearby a treatment center. Then this cell and gene therapy comes after no earlier treatment option when there is no alternative for the patient. So the patient are quite often in a very advanced stage of the disease. They might be very sick. And after eligibility, of course, when a patient wants to participate, he wants to have the treatment as soon as possible, but it's still not sure that he will get the treatment because there is a long process before the patient actually receives the treatment. So there is multiple barrier here. There is the patient level, because also for many patients, it's not an easy choice to say that he or she will receive a cell and gene therapy. And of course, there is a progression of the disease that might be a blocking factor for the patient. There is some site level restriction with referral to treatment center, to the fact that the treatment center are far from the home of the patient. And then there is a long-term follow-up, which is a regulatory requirement to follow up the patient up to 15 years. And here, what could be an opportunity? Maybe digitalization of clinical trial could help here, because it will allow to have a better and quicker information to the patient and also to the sites for extra training, with a way to support the sites, even if you're not physically here. And also the use of digitalization and decentralized clinical trial might definitely help for the long-term follow-up of the patient up to 15 years after the treatment. I wanted to conclude this presentation with a quote from John Adams, that every problem is actually an opportunity in disguise. And I truly believe that it's actually what happened with cell and gene therapy. There is a lot of challenges, a lot of creativity is often needed to transform this challenging into opportunity. And I think it's really true in the setting of cell and gene therapy. Some key takeaway before we go to the discussion on how to manage the clinical development for cell and gene therapy. My first advice would be to communicate early and constantly with the regulatory authority. Again, the regulatory authority are here to help, and they're happy to be with the company all along the way to build the product step by step. Then think about adapted study design to really meet the requirement of your study, of the indication of the patient. Regarding the logistical challenges, work with specialized provider and with that have true experience in this area, because not every carrier and provider can manage the complexity of cell and gene therapy. Then for the site and training of the site, ensure good training and good support for the site, because they need the company and the sponsor to be really here and to support the training. And for the patient, maybe digitalization of clinical trial might be a good way to improve the follow-up, improve the training, improve the knowledge around cell and gene therapy. And I think the key is always to keep an open mind and to be creative and collaborate with experts in the field. So there is a lot of ongoing. The question we can ask, and that's going to be open for discussion, is what could we do to do more? I think networking is key, of course, because if we share experience all together, we can find new opportunity and new way to answer the challenges. More training and more communication. Thank you very much for your time here. If you want to continue the discussion after this webinar, do not hesitate to follow us and to contact us on LinkedIn. Thank you.
Thank you very much, Virginie, for this fascinating overview. It looks like you took time to rest and to see the way how this kind of innovative therapies are managed and the way how to allow creativity be present in all steps, all such developments. So I have some questions that are already sent in the chat. The first one is raised by Tabuz Mohamed from KUL, I think, in Belgium. What type of digitalization can be incorporated during the clinical stage of ATMPs?
That's a good question, because it's a huge topic to talk about digitalization of clinical trial, and more specifically for the clinical stage of ATMP. I think the first thing is to help with the follow-up of the patient, because with ATMP, you have to ensure short-term, mid-term, and long-term follow-up of your patient. And patient retention is definitely a key subject to get enough data for the trial. So if you have decentralized clinical trial where the patient can be seen remotely and maybe have a blood sample in a laboratory nearby his house, it will definitely help with patient retention and to increase the follow-up of the patient. I think also digitalization can help having a strong training interface to be able for all the clinical site and also for the patient, by the way, to be accessible 24/7. Because I think there's a lot of question around the cell therapy, the way the cell are prepared, what are the adverse events, and many questions that both the site and both the patient might have. And having this kind of platform available 24/7 with videos, with information, I think is very important. This is two kind of way I think that could help with clinical trial in ATMP.
Thanks, Virginie. Second question that is coming from the chat from Fadwa Samsamani from Tangier. Why can't there be a worldwide regulatory authority that can make global regulations?
That's a very good question. It would be much more easier, definitely, to have something unified. But I think the good news is I think the regulatory authority are really trying to have more harmonization. There is some, I know there is some meeting between FDA, EMEA to get this kind of harmonization, because they realize that specifically for cell and gene therapy, this is a blocking point. And I think they are aware, and I think that this is also communication and participating with the regulatory agency would help to have the mindset change on their behalf and to change the regulation. But of course, changing regulation is not something easy.
Question for you coming from Bilal Mansouri. Okay, let's please go ahead. Bilal.
Hi, I think it's Malika Al-Mansouri. Sorry.
Sorry.
We have the same name. Yeah.
Sorry.
Yeah, it's okay. Thank you very much, Virginie and Mustafa, for this talk and this opportunity to attend. Actually, I have no experience in gene or cell therapy, but I have experience in regulatory affairs. Okay. And my question is really related to Morocco and the situation with regard to clinical trials for gene and cell therapy. Do you have some information to share? And what would you recommend, knowing that, or at least from what I know, is that the clinical trial environment is not well developed in Morocco? What would you recommend to progress in this field? Thank you very much.
Thank you, Virginie. Your answer? No, no. It's a virgin area. It is one of the reasons why the Moroccan Society for Regenerative Therapy has been created. The aim is to work on that landscape and nothing is done yet. And of course, this is an opportunity because in US, in Japan, in Europe, there are many lessons to learn and to use as a basis for the establishment of something that could be specifically dedicated to countries like Morocco. So there are many still things to do. Okay.
Thank you very much.
Thank you. Another question from Nabil Haji, please. Nabil, you are on mute.
Hello, are you speaking to me?
Can you hear me now?
Yes.
Nabil Haji?
Okay.
Yes. Excellent. I've been muting myself. Well, thank you very much for the great topic. Actually, this topic got many aspects because it's a multidisciplinary topic. Just my point here, I mean, we know that cell and gene therapies, while promising for treating various diseases, raise several concerns. I think you already have highlighted some of them as a key regulatory issues. Safety, efficacy, ethical compliance. If we can just mention one of it is the off-target and the long-term immune system response. As we know, I mean, toxicologists they are involved in any drug development or any new therapies. What we know, I'm Nabil Haji. I'm a senior toxicologist, but also a senior lecturer at Imperial College London. So I haven't introduced myself. So basically, the handicap is on the toxicologists. Sometimes they are not well prepared to provide a precision or precise risk assessment. My question, could the in silico or the AI, for example, could help partially the prediction of toxicity of the cell and gene therapy? If yes, how we can collate the data and how we can really organize the data based on separated or different technologies used and the model used. I think that's will speed up also the approval of the FDA on all the clinical trial committees, the ethical committees, and all that, if we have really strong prediction of the toxicity. Because that's actually the main gap, because we don't have, as a toxicologist, we don't have a data for that. Thank you very much.
Thank you, Nabil. Virginie?
Well, I think you might be better able to answer, but one topic, I think use of AI will be definitely also a key that's gonna change a lot of things in the development and speed up things. And again, I think regulatory bodies will be quite open to this technology if it's well presented and with a proper risk assessment behind that. So I think, yeah, it might be a key in the future to be able to increase the knowledge and to speed up things.
What I can add, Nabil, is that we are dealing with viable entities.
Okay.
So I think what those entities will behave depending on the environment you will apply on them. And of course, I think one of the answers that have been provided earlier on the multiple mechanism of action is that those entities will behave completely different in vitro and in vivo, because we will not be able to 100% mimic what could happen in the in vivo configuration, which could also be different from one patient to the other. But we have to address this question in silico,
yes,
in vitro,
yeah,
to at least, because we are still compiling data. I think all the cell and gene therapies, I mean trials both at the preclinical and clinical levels, we are trying to compile to understand, for example, how stem cells is behaving, how the new associated virus is behaving, and those we are still in early phases of learning and understanding. And this is one of the major reasons of the failure of many of earlier clinical phases.
Yeah, I just can add one thing here. I think this is a great opportunity to discuss with OECD and to define what are the models, as you said, in vitro, in vivo, that can be used for everyone. Then when you have a data, you can trust the database because everyone is using the same models. So I think that's the discussion need to be held as soon as possible.
Yes.
Yeah.
Yes.
Thank you very much.
Thank you, Nabil. There is one question in the chat raised by Ezil, his colleague from India, from Chennai. I think it is the same question raised earlier on the globality aspects of regulatory bodies. There is another question by Natalie. How are you, Natalie? So the question is the following: how can we effectively leverage the expertise of CROs and including CRAs to ensure that study design, the challenges are well understood and effectively communicated to investigators, other than classical protocol training, product training, or whatever?
I think I think it goes with what I said before, to say you have to work with provider who has expertise in the field. And by definition, if you consider the number of ongoing trials in cell and gene therapy, there is no that much CRO that really have expertise in cell and gene therapy. So that CRO quite often applies the classical rules and challenging that you have in classical drug development, and don't implement specific rules for cell and gene therapy. So I think I'm not aware of a CRO who is fully specialized in cell and gene therapy, but I really think that the best way to leverage this is really to have a contact with a CRO with a true expertise in cell and gene therapy, not only 1 or 2 trials sometimes, really a true expertise with a good presence on site, with CRA that are close, able to travel quickly and to go on site very quickly, to be there, because I think you need to have to show the sponsor support and support to the site. It's not only training, it's support to be there. It's be there physically, it's being there by phone or via video, but it's really being there for the sites.
Thank you, Virginie. A question raised by Adil Akush. Please go ahead.
Yes, thank you for a nice talk today. I'm Adil Akush. I'm associate professor at Western Michigan University Homer Stryker School of Medicine. My question is about the patient that you're using. So basically, you mentioned that they are sick, the only patient that you can harvest cells from them. So I'm curious about the quality of those cells and which type of cells you are harvesting. So are they stem cells or bone marrow? Then they need to go through like differentiation. So what is the steps that you're doing from the extraction of the cells and then the
well, actually, the example I gave was a more general example for the journey of the patient in an autologous setting. I cannot really answer to, because I'm not working for a cell therapy company anymore currently. So I cannot answer to the specific process. But yeah, I think the key is really to say that it might be a very long journey for the patient. The example was in an autologous setting. In a non-autologous setting, the process might be a bit quicker, and then the logistical challenge are bigger. But it was, yeah, just an example.
Now, what I can add on that question, I mean the selection of the product itself. If we will focus on cell therapy, we are looking to recover the altered tissue, the altered organ, by allowing the exogenous cells to be part of the diseased organ, so to try to reach a normal, or let's say a clinically accepted functionality of the organ, or we have to deal with other parameters that are, for example, impacting indirectly the organ, like inflammation or whatever. And the way how the cell type or the product itself will be selected depend on the indication, but also the readout you will use to evaluate the efficacy of the products you are using. So, and based on that, there is also another point that I think has not been raised by Virginie, is so far when we are talking about these innovative therapies using viable entities, we are not quite clear on how the dose can be considered. What is the number of those viable entities that can be used for getting a clinical effect? And this clinical effect could be short or at medium or longer term. And the way how the process will be applied would be completely different. Okay, another question from Hanin from Palestine. So the question is, what I will try to summarize in places like Palestine or other countries, how those innovative therapy can be efficiently implemented.
It's a very good question. I think it goes also with communication with the regulatory bodies to be also able to provide this therapy to the patient. And it brings, of course, some additional challenges, but that's a very good question. And I think the awareness of this specific disease with time, it's growing and growing and growing. And I hope with the time, it will be very an international awareness, and that this treatment will be able to be implemented everywhere in the world.
Yeah. Another question, Virginie, from Taboos, also Mohamed. How do you view the variability between donors, and how do you account for it during the clinical stage?
Maybe you can also answer with that, Mustafa. You have maybe more experience on that.
And this can be addressed case per case, depending on the type of the cells, depending on the percentage of cells you will get from the raw material tissue. It's a case per case, I think.
And that's also actually one of the challenges, that two therapies might be totally different on some aspects, and there is no general answer, unfortunately.
Professor Younes Benfdil is asking is there some effort to simplify the techniques?
I think there is. I think that every process techniques and manufacturing aspects are trying to be simplified along the development. And it's also accepted by the authority that you might have a process in place at the early stage of your trial that might evolve later and change a bit later during the clinical process. I think there is actually some strong effort to simplify, because if the company is targeting to go on the market and to target a large population worldwide, at some point you have to have a process that is viable and as simple as possible to be actually effective on the market.
Thank you. Another question from Saida Rahmani. Please go ahead. Saida, yes.
Hello, salam alaikum.
Salam.
I would like to thank you for this immensely. I would like to thank you immensely for this interesting topic. I'd like to thank you for the invitation. I would like to thank the speaker and the organizers. My questions revolves around the challenges met in countries like Morocco, specifically patient-wise. How do we convince patients to enroll in clinical trials you know, the limitation in understanding and awareness about the use of gene and cellular therapy, specifically in Morocco. Thank you.
Yeah, I think it's a very important topic to increase information of the patient, because cell and gene therapy can have a very also bad reputation due to some abuse in the past. I think here I cannot answer for Morocco specifically, but I think it applies also to any other country, is again, increase information to the patient and to provide the information to the patient to explain that what does it mean to receive a cell therapy or a gene therapy. I think it will take time, because unfortunately the abuse and some bad things that happened in the past leave a strong impact in the mind of the patient. But I'm pretty sure that with extra information and extra training for the patient, hopefully it will change in the future, but it will take time, definitely. Mustafa, maybe you have some comment there.
It's the truth, but of course, the way how countries, developed countries like US, like several in Europe and in Asia also, we have to learn from all what these countries did perform, because those countries also start from scratch. So we have to learn how to deal with such development. And also what could be very important is try to adapt the process and the system to the country configuration. And this is dependent on the way how the physician or the people, the professional that are implicated, how they will learn and how they can transmit their knowledge, because it is a matter of knowledge and transmission of the knowledge. And of course, this is the first limiting step. The second limiting step, and this is one of the particularity of these innovative therapies, is the multidisciplinarities. So the physician has to talk and to collaborate with the researcher, with the regulatory bodies, with the people specialists in the ministry. And this is something that is not obvious for the other, therapeutics, by using drugs or even surgical intervention or whatever. You need the radiologist to implement the catheter. You need biochemists to evaluate monitoring or whatever. And this is a kind of a new mind of working. Unfortunately, in our countries, we are not yet there. And it is also one of the limitations that can hamper an easy implementation of such kind of innovative therapeutic. It is not a problem of expertise or accumulated experience. It is a kind of a vision and a voluntary expectation. I will try to compile the comments of Dr. Rashid Abuzid and Rosanna from Mexico also. Both questions are related on the way how governments, international organizations, and healthcare system, how they should work to make the use of such innovative therapies real.
I think there is already some work ongoing. I know there is some common meeting between some region, at least, maybe not on a country level, but some on regional level, to try to harmonize and work on the regulation. But I think it should come also from the company, but it's a never-ending circle, because some company doesn't want to go in the country if there is no clear regulation there, but then the regulator would not question themselves and change the regulation if the product is not there to develop. So I think it should also come from the company itself to say, let's try to go in this country because I know some great clinical sites that are open to this therapy and are willing to. And I think then it can change the mind of the regulator. I have a personal story here very shortly that we tried to, with the previous company, Celayan, to go to Argentina. And actually the regulatory body was not aware of how to deal with this kind of therapy. So it took longer because they had to go to specific regulatory body. So it took longer, but I think it might have changed the way and the perception of cell therapy, and maybe it will change later the regulation. But so I think, yes, the regulators should talk together, but I think the company should also try and say, I want to go there because the patients are there, because the clinical sites are willing to do that. And I think at some point the regulatory body will follow.
Adil, for you, you're on mute.
Yes, thank you. Yeah, I just want to add about the implementation of such like clinical trials in Morocco all around the world. So here in the US, I want to speak up about the US. We have the same problems. So we have, even with the advancement that we have, we still have limitation in recruiting some type of populations. African Americans, we can't recruit them. Latino, we can't recruit them. Immigrant from Middle East, we can't recruit them. And what we are trying to do now here is to work with the community. So we have like small community.
Sorry, you're on mute again.
It's mute by itself. So yeah, I'll keep an eye on it. Yeah, so basically we have our department for clinical trials, and we have people that they reach out to the community and talk to them and explain to them the safety of the product that we want to use for them. So basically it's a win-win collaboration. So basically we collaborate with them, and also the societies. So basically the cancer societies or infection societies. So people trust those societies. And I'm very glad that we have a Moroccan Society for Regenerative Therapies, which englobe a lot of things in there. So gene therapy, cell therapy, tissue engineering, and biomaterials. And basically this is a good way to start. As Dr. Najimi mentioned, we need to learn from them. So we need to learn from the other people, and we need to learn from their mistakes. So why we have this people that they don't want to get into this clinical trials is, for example, the HeLa story about, the HeLa cells that everyone is using in their labs. So they were isolated from an African American without a consent. So, and they still used until now. So basically the consent is very important, and explaining and collaborating with the patient through the hospitals, through the government, through the communities is very important to start like a nice, background, or clean and clear background to start clinical trials. And specifically for cell therapy is very different. Here we have some clinics that they inject PRP, that they inject stem cells for like orthopedic patient, but there is no science behind that. So, but basically they still use it.
Yeah, this is a topic. This is a very good topic that should be addressed. And Morocco is really concerned by this, for example, kind of interventions, including PRP. And I think we have to organize a meeting on that aspect. I just want to say one word, just to say that we are, as a Moroccan Society for Regenerative Therapies, we are really honored to have with the participants, Professor Shur Eric, who is the CEO of Hepatex in California. And he is also a colleague in the GI committee of the International Society for Cell and Gene Therapy. And really, I would like to have his word in this meeting. Please, Eric.
Thanks, Dr. Najimi. It's really been a terrific seminar. I've loved hearing all of the comments, and what I really like is that this discussion is kind of going on worldwide right now. As you mentioned, you and I have a really strong mutual interest in GI disease and trying to bring something to patients with liver disease, which is it's an enormous problem globally. And I was actually at a conference yesterday in California, talking with a group of folks about how can we accelerate these clinical trials, because it's really the key leverage point where value is created, and if we create value, then we'll see resources flow to this area and the process will accelerate. And so Virginia, thank you for pointing out some of those things that we talked about yesterday. Adaptive trials are one. I think very careful selection about who you work with to develop these products is important. I think Dr. Najimi knows a lot about that. So we're doing our best to bring something forward for these liver disease patients, and happy to share with others in the community to mutually make this go faster.
Yeah. Thank you very much, Eric. So
Thank
I think there is no more question as I'm seeing here in the chat. We are respecting perfectly timing, 4 minutes, besides the 60 minutes originally allocated to this webinar. So my summarized view on all discussions, first is learning, transmitting the knowledge, discussing deeply between physician and non-physician and regulatory bodies, ministry agencies, and of course, adapting each configuration to each disease, to each country, which could be, we don't speak about the religious aspects, we don't speak about other indication that would be potentially the subject of the coming webinar. So I would like to thank you all for your participation, for your active participation. Thank you all the colleagues that join us from around the world. And we were happy because I think at 5:32, we were around 82 people that were joining the webinar, which is quite important. And also I can see from the audience, many students that are interested by this kind of innovative therapies. My last word is for Virginie. Thank you again.
Thank you
for your collaboration, and for my friends Simo and Najib. Jessica want to say something? No? I see your hand.
It was just applause, not the hand raise. It's just the applause.
So thank you very much.
Thank you.
Enjoy the end of the week. Thank you.
Thank you. Bye-bye.